Yes, I'm about ready to start a video. You want up here? I'm gonna start a video. I know, I'm starting a video. That's what I'm doing. I'm trying to start a video. Do you want up? You want to say hi? All right, come on. Oy. Come on, baby Tig. Hey everyone, it's baby Tig. He's here to say hi. All right, you can join me for the... Oh, he's purring. Do you guys hear him purr? He's here to join me as I introduce today's topic, Chapter 11. Let me situate you guys so you can see him a little better, maybe. I think you could see him now. Welcome back, you guys. It is time for Chapter 11. We'll be covering that, characterizing and classifying prokaryotes. Welcome back. Let me give you guys a little better angle on little Tig. There we go. Hey Tig, you gonna join me? All right, that's fine. So chapter 11, we're talking about prokaryotes, and we're starting with the Archaea. Remember, Archaea are a whole domain of life, and they include prokaryotic cells. They are comprised of prokaryotic cells, and there are two phyla. Remember how taxonomy works? There are kingdoms, but you know, there's actually three domains of life, I should say. There are three domains of life: Archaea, bacteria, and eukarya, right? So the Archaea are one of the three domains of life. They are comprised of prokaryotic cells. And if you think about it, we don't talk about them much in medicine. Well, and there's a good reason for that. The reason is because they don't tend to cause too many human diseases. So they kind of get neglected in the medical realm. So we're not going to spend a ton of time talking about the Archaea, but we are going to touch on them. And what you need to know for this chapter is you need to remind or refresh yourself as to how taxonomy works. Do you guys remember King Philip came over for good spices? King for kingdom, Philip, phylum, came, class, over is for order, for family, good means genus, and spices for species. Okay, so remember that that taxonomy scale, because we're going to be talking about that a lot in this chapter. So if you look at this, Archaea are best known for growing in anaerobic environments, hypersaline environments. That means very salty environments. They can grow in pH extremes. They could be acidophiles. They could be alkalinophiles. They can be thermophiles, right? They can grow in high temperature habitats. So basically, Archaea are best known for growing in extreme environments, places where humans would not want to venture for the most part. They can even be found in aquatic environments, but also Arctic temperatures, so really cold places as well. So just keep in mind that because Archaea can grow in these extreme environments and prefer to grow in these extreme environments, they typically do not cause human disease. Because remember, I made a big point about this earlier this semester, and that is that the reason why certain bacteria cause disease is because those bacteria prefer to grow on the human, right? So mesophiles, neutrophiles, these types of bacteria prefer to grow on humans, okay? These Archaea don't cause diseases in humans very often because the human body is not an extreme enough environment for growth, okay? So that's kind of how it works there. So let's talk about the taxonomy of Archaea. So what you need to understand is that there's the domain Archaea, right? Domain. And then there's the kingdom Archaea as well. Now, what you need to know is that there are two phyla under that domain and kingdom. The two phyla are called Archaeota. You see here, let me show you. Archaeota And the other phyla of Archaea are called the Archaeota. Okay, so let's talk about the Archaeota first. The phyla Archaeota. These are extremely thermophilic. Remember what that means. That means they grow in very hot environments. I'm talking 85 degrees or more environments, boiling habitats. Hyperthermophiles can live in these hydrothermal vents, like the sulfur cauldron of Yellowstone National Park, for instance, where it's full of sulfur and it's bubbling hot. If you were to fall in there, that would be a quick demise for any human or any animal that were to fall in there. Most are anaerobes. Most are acidophiles. They're sulfur dependent. They require sulfur. So they're probably lithotrophs. Remember, lithotroph means requires sulfur as an electron for electron source. Now, the Archaeota, on the other hand, the Archaeota are much more diverse, right? Some of them are methanogens. They produce methane. Some of them are halobacteria. They require high salt. Some of them are thermoplasms. These are the thermoacidophiles. They grow in high temperatures. Some are extremely thermophilic sulfur metabolizers. Some are sulfate reducers that use sulfate as an electron acceptor. So you can see here that there are many different. Hey, he's back. And there are many different groups of types of Archaeota. Oh, he doesn't want up. He just wants to yell at me this time. All right, Tig. Hi. All right, he might want up later on. You guys can see him later on. Right now he's playing. So anyway, what you need to know is that, again, Archaea don't typically cause many diseases. Why is that? Because they don't prefer your body's environment for growth. There are two phyla of Archaea to know about. The Archaeota, which are very restricted. Look at this table here. Look at this table. These are the groups of Archaea, what they do, and these are the representative genre, right? So look at this. Notice this. You see the genre that have a C? I'm highlighting them here, the ones that have C. Those are the Archaeota. Notice that the Archaeota are entirely extremely thermophilic sulfur metabolizers. However, look at the other genre, the ones that say E. Those are the Archaeota representatives. Notice how there are Archaeota representatives in each and every one of these groups of Archaea. What that should tell you is that the Archaeota are quite diverse, whereas the Archaeota are not. The Archaeota are like one-trick ponies. They are only extremely thermophilic sulfur metabolizers. Hey, Tig. Howdy. Tig makes sure I take a break every 2 minutes. And he doesn't want up. He gets my attention, then he runs away. Probably because he wants food. But he just ate. All right, so let's keep going. Let's talk about bacteria now. Let's move from Archaea. Hi. Let's go from Archaea to bacteria now. The bacteria we're going to be talking about are the phyla Dinococci, as well as the non-Proteobacteria gram-negatives. These are different phyla of bacteria now. So let's talk about these bacteria. Look at all of these phyla here. Okay, let me show what I'm talking about. Go back here. Look at this. So we already talked about the Archaea, which are the Archaeota and the Archaeota. Now we're talking about all of these here. All of these are bacteria. And the ones in color are the ones we're about to discuss. You see, the ones in color are the ones we're about to discuss. These are known as the gram-negative non-Proteobacteria, right? You see this? These are the gram-negatives that are in color right here. These are phylum of gram-negatives. In fact, the Proteobacteria are also gram-negatives, but we're not talking about them yet. The only two phylum of bacteria that are not gram-negative are these two. Look at this. the low GC gram-positives and the high GC gram-positives. These are the only two phyla of gram-positives. The rest of these are gram-negatives, including the Proteobacteria. But we're not talking about the Proteobacteria yet. We're talking about the gram-negatives that are non-Proteobacteria. Okay? I know that's a mouthful, but try to make sense of that. So we're going to start talking about these. We're going to go phyla by phyla by phyla, and we're going to talk about each phyla. All right? So we're going to start with what? Aquaficiae. Let's start with this one. Aquaficiae. So Aquaficiae, what do I want you to know about them? These are the oldest bacteria. These are the most ancient bacteria. All other bacteria kind of branched off from a common ancestor of these bacteria. They tend to be thermophiles. Okay, so that's really all I really need you to know about Aquaficiae. They tend to be, they're these ancient, old creatures. They tend to be non-aerobic, right? They tend to be, they cannot live in oxygen for the most part. Okay, and then we've got the Thermatogae. Let's talk about the phyla Thermatogae. Phylum Thermatogae. This is the second deepest branch, all right? So that's all I need you to know about those two phyla. Otherwise, they aren't really known much for causing human diseases or causing human problems. So we're not going to delve too deeply because we could really get lost in the rabbit hole of talking about creatures. There's thousands upon thousands of different creatures we could talk about and discuss, but I'm trying to do only a cursory overview of the ones that are less important for medicine, and we're going to delve more deeply into the phyla that are more interesting in terms of, your career as a nurse or a practitioner. So what's next? We've talked about the Aquaficiae. This phyla is the oldest branch of bacteria. Then we talked about Thermatogae. These are the second oldest branch. And now we're off to the Deinococcus thermus, right? Deinococcus thermus. What you need to know about this phylum is that they are extraordinarily resistant to desiccation and radiation. They are weirdly resistant to desiccation and radiation. What's desiccation mean? You should know that desiccation means from drying out, right? Drying out. So these are resistant from dry conditions. Most bacteria prefer a moist environment, or they need some access to water. However, the Deinococcus thermus, these are more resistant to desiccation. They are also more resistant to radiation, okay? So that's what I need you to know about these guys, the Deinococcus thermus. Let's talk about the phylum Chlorobi. These include your green sulfur bacteria. These are photosynthetic bacteria, the green sulfur bacteria. Okay, so look at this. Where did we just talked about Chlorobi here. Okay, let's talk about the phylum Chloroflexi. These are the green non-sulfur bacteria. That's all I need you to know about those. Okay, now let's go on to some more interesting phyla where, they have more of an impact on humans. How about the phylum Cyanobacteria? The phylum Cyanobacteria. Which one is that? Here, the phylum Cyanobacteria. Do you guys know what cyanobacteria are? They are photosynthetic bacteria. Photosynthetic bacteria. So they undergo photosynthesis. They take CO2 out of the air and they fix the CO2 and they undergo the process of photosynthesis. So they convert the CO2 to sugar and other organic molecules. They also produce a lot of oxygen. So the reason cyanobacteria are important is because they are a huge group of photosynthesizers on the planet. They fix CO2, which means they remove CO2 from the atmosphere, and then they also create oxygen for us to breathe, and they produce sugars and other organic molecules that we then ingest as heterotrophs, right? Next phylum. This phylum is very interesting. This is the phylum Chlamydiae. These are obligate intracellular parasites. Obligate intracellular parasites. So these are bacteria that actually are parasitic and live within your host cells. They grow and reproduce inside host cells, and humans are the host. These cannot create their own sugars. They cannot catabolize carbohydrates. Sorry, not only create, because animals can't create carbohydrates either. They can't break down sugar. So they don't have a way of breaking down sugar. They don't have a way of making ATP. They don't have a way of regenerating NAD+. So do you know what that means? This is why they're obligate parasites. They require your ATP from your cells. So that's why they're parasitic. They're using your sugars. They're using your ATP. They're using your NAD+. They are draining your cells of energy. And of course, you know that this is the genus Chlamydia. This is the causative agent. This is the bacteria that causes the STD, the sexually transmitted disease, chlamydia. Here's another STD causer, syphilis, that causes syphilis. It's an example of the phylum member Spirochaetes. Spirochaetes are long, slender, and have a helical shape. The reason they have a helical shape is because of that axial filament. We discussed axial filament in the past. Remember, that's a structure that allows for motility, corkscrew motility. So a lot of these helical bacterium, their motility, their mechanism of motility is corkscrew. They are able to corkscrew through mucus and other thick viscous fluids, okay? And they can cause diseases, like in humans, like Lyme disease, syphilis. These are spirochete diseases. However, not all of them are disease causers. Some of them are symbiotic with termites, symbiotic with various mollusks or mammals, in the oral cavities of animals. So they're not necessarily all bad, but a lot of them do cause human disease and suffering. Okay, so that was an entire... So what did we just cover there? Again, we covered all of these phyla that are in color here. See, Spirochaetes, Actinobacteria, Chlamydia, Corobi. We didn't talk about the Bacteroidetes. We didn't talk about those much, but we touched on most of these gram negatives that are the non-Proteobacteria gram negatives, right? Now, we're going to touch on this group here. This is actually a massive group here called the Proteobacteria. We're going to talk about the Proteobacteria, the phyla Proteobacteria. And by the way, these are gram negatives as well. So that's where we're at now. Chapter 20, the Proteobacteria. So the Proteobacteria include beta Proteobacteria, gamma Proteobacteria, alpha, epsilon, delta Proteobacteria. These are all Proteobacteria classes. So what you need to understand is the phylum Proteobacteria are gram negative, and there are 5 classes of Proteobacterium that we're going to talk about. So let's talk about them. Again, the alpha Proteobacteria, the beta Proteobacteria, gamma Proteobacteria, delta Proteobacteria, and the epsilon Proteobacteria. These are types. And I have another visitor. It's Little Tig. Hey. So those are the classes of Proteobacterium. Okay, so let's start with alpha Proteobacterium. Do you finally want up? Or are you going to run away? You're going to run away again. One of these times he'll want up and say hi to you guys again. So again, this is the alpha Proteobacteria. Most are oligotrophic, which means they grow with low nutrient levels. They don't need high nutrient levels to grow. This is why you can find them in the oceans, where there might not be tons of resources, tons of nutrients around. Okay, some can have evolved to live within plants and animals. So a lot of times you can see these as parasitic as well, like intracellular parasites, right? So for example, Rickettsia. Hi, Tig. Rickettsia. Rickettsia are intracellular parasites. They use the host's ATP. right? Just like chlamydia did, right? They use the host's ATP. So for example, here's some bad examples here. Rickettsia prowazekii and Rickettsia typhi, which causes typhus fever. Rickettsia rickettsii. This one causes Rocky Mountain spotted fever. So you can see a lot of these bacterium are intracellular parasites. Their genome sequence is similar to mitochondria. So they live within your cells and they use up your ATP and they cause damage. Okay, and they're spread a lot of times by blood-sucking insects. If you wonder how you catch these, they're spread by blood-sucking insects, such as ticks. Ticks can spread them. The tsetse fly can spread these. You want up? Come on, Tick. Come say hi. Wait, here he is, guys. He's back. Hi, Tig. All right, so let's keep going. Here's another family member here of alpha Proteobacterium. This family member is called the Colobacteriaceae and the Hyphomicrobiaceae. I know these are tongue twisters. These are marked by the presence of a prostheca. Okay, prostheca is a stalk that allows these. Oh goodness. All right, there goes all my slides because Tig decided to, I'll show you, he decided to lay all right on the keyboard and that completely got rid of the slides. So sorry about that. Let me get the slides back up. Sometimes we just need Tig's comical intervention. That's not it. Swap. Where were we? Gosh. Oh, Tig. Tig doesn't make it easy sometimes. Okay. Here we are. So again, we're talking about the prostheca. These are stalks. These are extensions of the cell, including plasma membrane, and they allow for attachment. The stalk will allow for attachment of the cell to a surface. And also a lot of times you'll see these reproduced by budding. So not a type of binary fission, but progeny is a bud that first appears as a small protrusion on the parent cell and enlarges to form a mature cell, such as seen in eukaryotes like Saccharomyces, like yeast. You'll see budding in yeast as well. So let's move on to the next class. These are called the the beta Proteobacteria. These include the Neiseriales, the order Neiseriales. The genus you might be interested in here are called the Neisseria. Okay, these are called the Neisseria. And with Neisseria, this is another type of human pathogen. Okay, these guys are human pathogens. They cause gonorrhea. So Neisseria gonorrhoeae causes gonorrhea. Neisseria meningitidis causes meningitis. Okay, and the reason they're hard to treat is because they've got capsules, right? Capsules help to cloak the bacterium from the host's immune system. So let's move on to gamma Proteobacteria. The gamma Proteobacteria are very diverse, so we're going to talk about some of them. They include the order Methallococcales, which includes the methallotrophs. You know, the methallotrophs are bacteria that are very useful in our fight against global warming. These bacteria use one-carbon compounds. For example, they will use methane. Think about a bacteria that can take methane from the air and use it as the sole source of carbon. So these are very handy bacteria. I could see that these bacteria might be useful in our fight against global warming in the future if we could grow more of them, or if we could utilize their metabolism somehow. The next order, Pseudomonads, right? The Pseudomonads in the order Pseudomonadales. These are very important. They degrade organic molecules. They're involved in this process called mineralization, which means that these organisms break down organic materials into inorganic substrates. So they're recyclers. They're recycling molecules in nature. The next order that's interesting for you guys, and also interesting because they cause so many human diseases. This is the order called Vibrionales, right? These are the Vibrio, the bacteria Vibrio. Right, Tig? Tig agrees. The order Vibrionales, these are, you remember what Vibrio shape means? It means curved shape, okay? So these bacteria have a curved shape to them. They look like a comma, right? And they include the genus Vibrio, and the species is Cholerae. These are pathogens that cause cholera. By the way, I messed up here. Let me reach around Tig real quick and change this to a little. See, it's hard to type. Oh gosh. Hold on, I'm trying to fix a slide. All right, it's fixed. Let me show you what I fixed. There. I made a lowercase c here. You see, Vibrio cholerae. Remember, the proper way of writing binomial nomenclature is capital on the genus, lowercase on the species. The next order that we'll be talking about are the Enterobacteriales. These are known as the Enteric bacteria or the Enterobacteria. Okay, these are facultative anaerobes. You remember what that means? That means they can grow with, in the presence or absence of oxygen. These include members such as Salmonella, which you have heard about, that causes a lot of human diseases, Shigella, which causes dysentery, Klebsiella, which causes pneumonia, Yersinia, which causes the plague. Okay, a lot of these members cause human suffering, human diseases. So, because a lot of these can be diseases or bacteria that grow on humans. Now, one thing you should realize is you have a big unknown report this semester, correct? Remember the unknown identification report from lab? The family member that your unknown belongs to is the family Enterobacteriaceae. Enterobacteriaceae, which includes well-known bacterium such as E. coli. All right. Oh, it looks like Tig might just be on his way out. All right. Welcome back. Guess I'll fix my... There we go. There we go. Perfect. We're back. So the family Enterobacteriaceae is the family of your unknown. This is a huge family of different genus of creatures, and E. coli is the most famous one. E. coli is actually the most best studied bacterium of all. It's found as a normal inhabitant of intestinal systems of animals, including humans. In fact, if you see E. coli anywhere, it's indicative of fecal contamination. So, for example, if you saw E. coli in a stream, it has no business growing in a stream. Its reservoir is not the stream. The way the E. coli got to the stream was that it contaminated the stream with feces, right? So wherever you find E. coli outside of the intestines, it's a indicator of fecal contamination. They're known as fecal coliforms. Okay, so anyway, some strains can be pathogenic. E. coli. The mainstream strain. I'm sorry, I'm getting all jumbled up here. The main strain of E. coli is non-pathogenic. You have E. coli in your intestines. I have E. coli in my intestines. E. coli is not a dangerous bacterium. However, there are strains of E. coli that can be quite dangerous. These can cause gastroenteritis, UTIs. You can even get dysentery from a strain of E. coli called O157H7 strain of E. coli, which has picked up the Shiga toxin gene from Shigella. So you can find E. coli that are deadly, but you can also find E. coli that are not, right? It depends on the strain of E. coli. Let's talk about another class, the Delta Proteobacteria. We're moving on. What's really interesting here is this order Delovibrio. What's cool about the Delovibrio is that they behave similar to viruses. Let me explain. Here's how Delovibrio works. Delovibrio is a smaller sized bacterium, comma-shaped because it's a vibrio, and it's flagellated. When Delovibrio attaches to a bacterial prey, so like another bacterium, it will bore into that bacterial cell wall, embed itself inside of that bacterium, so it's effectively infected that bacterium. It will then grow in length like a party balloon, okay? Grow really long, and then it will undergo a process known as multiple fission, not binary fission, multiple fission. So it will actually start looking like sausage links, and then each one of those sausages would become a new creature, a new Delovibrio. They will then lyse the cell and find more prey to infect. So this reminds me, is reminiscent of bacterial, or I should say viral infection of bacteria. So this Delovibrio behave like virus, even though they are not virus. Next class, we're going to discuss the epsilon Proteobacteria. What you need to know is the genus example here of Helicobacter. What's interesting about Helicobacter? They can cause ulcers, right? So in humans, it was thought a long time that ulcers were caused by maybe stress or diet. Now we know the main causative agent of ulcers is Helicobacter pylori. It's a member of this genus, Helicobacter pylori. This bacterium grows in the inner lining of the stomach, and it can cause peptic ulcer disease, ulcers. It can also cause gastritis. All right, so now we have thoroughly taken a nice, sufficient overview look at gram-negative bacterium. We looked at the non-proteobacterium gram-negatives, and then we just finished our wrap-up of the proteobacteria gram-negatives. We have yet to even look at any gram-positives, and that's what we're going to do now. There are only 2 phyla of gram-positives, the low GC gram-positives and the high GC gram-positives. So let's get started with that now. Let's start with the low GC gram-positives, known as the gram-positive bacteria of the Firmicutes type, right? Firmicutes. So let me show you. Now we're talking about the gram-positives, the low GC gram-positives. What's another name for them? The Firmicutes. After we finish this, we'll talk about the high GC gram-positives. They're also known as the actinobacteria, okay? So let's start it with the Firmicutes, the low GCs. These include 3 classes. These classes are the Molecutes, Clostridia, and Bacilli. So let's talk about the Molecutes, also known as the mycoplasms. These guys lack a cell wall. Do you guys remember that I said that bacteria have a cell wall of peptidoglycan almost always? Well, these are the exception, right? You have these bacterium do not have a cell wall. They're still considered gram-positives because they don't have an outer membrane, but they don't have the peptidoglycan cell wall. And because they don't have the peptidoglycan cell wall, they're called pleomorphic, which means they don't have a particular shape. Think about it. If you don't have a cell wall, your cell is not held in any particular shape, so you're not going to have a particular shape to you. Okay, these also tend to be very tiny bacteria, very small bacteria, and because of that, a lot of them are causative agents of pneumonia. Think about it. If you're a tiny bacteria, smaller than a typical bacteria, well, then you're probably more easily aerosolized. If I cough, I cough, those little droplet nuclei floating out from my mouth, those can harbor these tiny bacteria, and those bacteria can travel further distances, probably down your respiratory tract, and my pneumonia can become your pneumonia via transport of these tiny pleomorphic bacteria through aerosolized nuclei, right? Droplets. Okay, let's talk about the next class, the class Clostridia. This includes their large genus of Clostridium. Clostridium are very fascinating bacterium because they are endospore formers. Do you guys remember earlier in the semester I told you that there are only two classes of bacterium that can produce endospores? There's Clostridium and there's Bacillus. Okay, remember that for the exam. There's Clostridium, which is these guys, and Bacillus, which is, we're going to talk about that in a little bit. Okay, so endospores. And remember what endospores are? Very strong protective shells where bacterium are, where the bacterium can preserve its genome so that later on, after the insult has passed, let's say UV desiccation, radiation, heat, whatever insult has passed, those endospores can then germinate and grow, and the bacterium can go back to a vegetative state. So, what do we know about Clostridium? They can cause food spoilage, Clostridium botulinum. Remember, I had that example of these Clostridium botulinum growing inside of my can of diced tomatoes. That would be an example of botulism, which is a disease caused by the Clostridium botulinum toxin called Botox, right? This Botox toxin is a neurotoxin, and when ingested can cause severe side effects up to and including death. Another example of Clostridium is Clostridium tetani. This is the bacterium that causes tetanus, another neuro disease that can cause uncontrollable contraction of muscles. All right, and then the next class, this is the class Bacilli, which are, again, the other group of endospore formers. Remember, there are only two classes of bacteria, two genus of bacteria that can make endospores. These are the Bacillus and the Clostridium. Bacillus can make endospores. Now, look at this. Bacillus can take it one step further. Bacillus can form antibiotics as well. So, what are antibiotics? Remember Alexander Fleming and his discovery of penicillin, I should say, the antibiotic penicillin from the fungi Penicillium. Antibiotics are chemicals that disrupt or destroy bacterium, right? These can be naturally generated. Remember, molds can form antibiotics. Certain bacteria can form antibiotics. So Bacillus is capable of producing antibiotics against different species of bacterium. Different genuses of bacterium would die from the antibiotics produced by Bacillus. Okay, so for example, Bacillus subtilis is an example of a Bacillus bacterium. It's an endospore former, soil dweller. And again, you should be aware that Bacillus are quite hardy little guys. Not only can they form an endospore if the going gets tough. Let's say the environment gets harsh. Bacillus can form endospores to ride it out. But if the Bacillus wants to grow and take over resources, it can produce antibiotics. So it can kill other species in the environment. So it can monopolize those resources for itself. It can take all the sugar source. It could take all of the nutrients by killing its competition. So as Dr. D, if I could somehow come back as a bacterium in another life or something, I would want to be a Bacillus, right? If I could be, those mind activities, mind exercises, like if you could be any bacterium, which one would you be? I would actually want to be a Bacillus because if the going gets tough, I can ride it out as a endospore. And then when I'm growing, I can decimate my competition with antibiotics and monopolize all of the resources. So that's why I say that I would want to be Bacillus. Okay, let's talk about a family member here called not Strepto, that's coming up soon, Staphylococcaceae. These include the Staphylococcus. These are facultative anaerobes, so they can grow with or without oxygen. And they're normally associated with the skin, right? Remember, because they tend to be halophiles, right? They tend to be halophiles. They tend to grow where salt is high. Okay, so let's talk about a couple. And again, I should have put a lowercase e here for this epidermidis, a lowercase a. I think it just got rearranged. But remember proper binomial nomenclature. I should have a lowercase e here, a lowercase a here. But anyway, let's talk about Staphylococcus epidermidis, skin resident. You can find this if in a surgery gone wrong, a wound infection, surgical infection, urinary tract infections. Again, this is a normal skin resident, which is a opportunistic bacterium. That means Staphylococcus epidermidis grows on the skin and normally doesn't cause any harm. However, if you were to get a wound, if you were to get a surgery that's not properly treated, if you were to introduce these bacterium to the urinary tract, well, then it could cause an infection and pain and suffering. And then you have Staphylococcus aureus. Again, this is a normal skin resident as well. However, it also causes pathogenicity. It's another opportunist. If you have any kind of break in your skin, well, then you can get staph infection. And staph infections are quite harmful because of all the virulence factors. You don't need to memorize all of these virulence factors that staph has, but these virulence factors are properties of the Staph aureus that make the staph particularly harmful as a pathogen. Okay, and now to make things even worse, we're finding more and more cases of resistant Staph aureus, antibiotic resistant Staph aureus. In the medical setting, you will come to learn about methicillin resistant Staph aureus, also known as MRSA, vancomycin resistant Staph aureus, known as VRSA. These are very resistant strains of staph. You do not want to get infected, like let's say a skin infection with MRSA or VRSA, because there may be no treatment. There may be no way of even treating the skin infection, and then you may have to resort to amputations and such. They can be life-threatening. All right, let's move on to another order, the order Lactobacilliales. These are the lactic acid bacteria, very, very useful bacteria, because you can use them for fermentation, right? They ferment sugars for energy, and we can use them to ferment all kinds of foods for us as well. So let's talk about lactobacillus. In dairy products, they're used to make meats, waters, beer, fruit, other materials, yogurt, right? They're normal flora of the mouth, intestinal tract, also parts of the body as well. Okay, so like I said, the lactobacillus are very, very useful in producing fermented products for us, vegetable products, sauerkraut, pickles, beverages including beer, wine, juices, milk, sourdough bread, cheeses, yogurts, sausages, probiotic agents, in beer, wine, milk, meat. Okay, so you find them all over the place, and they can also cause food spoilage. All right, so now we have discussed the low GC gram positives, the Firmicutes. Now we're moving on to the high GC gram positive bacteria called the actinobacteria. Let's talk about those, and that'll round out this chapter, right? These chapters. So let's go ahead and start. Actinomycetes. These are the high GC gram-positive bacteria. This is the final genus, I should say the final phylum of gram-positives we're going to discuss. These are also known as the actinomycetes, all gram-positive aerobic bacteria that produce filamentous cells called hyphae and differentiate into asexual spores. So let's talk about these. The actinomycetes, these are a source of a lot of antibiotics. These are bacterium that are known for producing a myriad of antibiotics. How do the actinomycetes grow? Their development, they form filamentous cells called hyphae. So they're kind of reminiscent of fungi. You remember how fungi grow? They have a filamentous shape. And then fungi form a mycelium, which means this network of hyphal cells. And then at the tips, you even form spores, like these non-sexual spores, right? They're not motile, but they're spores, just like fungi. So what you need to know about the actinomycetes is that they grow and behave an awful lot like fungi, like the eukaryotic fungi. They have a hyphal growth, they form a mycelium, they form spores, okay? Instead of growing by binary fission, they'll form spores, and they'll divide that way. Very strange, very interesting. And they form a lot of antibiotics like fungi as well. So they behave and act, and their biochemical pathways are a lot like fungi. However, they are not fungi. So let's talk about a suborder called Corynebacteriaceae. And these include the Corynebacterium, the Mycobacterium, the Nocardia. Let's talk about these guys. The Corynebacterium. What you need to know about these is that they are common human and animal pathogens. For example, if you've ever heard of Corynebacterium diphtheriae or C. dip, this is the causative agent of diphtheria in humans. This is why you get your vaccinations, to prevent that. Mycobacterium. These are bacterium that contain these mycolic acids. Remember that waxy substance in the cell wall? These creatures have cell walls that are waxy and impenetrable by antibiotics, making them very difficult to treat. So a lot of times if you have a mycobacterium infection, that could be lifelong, right? And it's because of that mycolic acid in the cell wall, making it very impenetrable to antibiotics. Remember, these are the acid-fast bacterium. These include pathogens such as Mycobacterium tuberculosis, which causes tuberculosis, and Mycobacterium leprae, which causes leprosy. Let's also talk about the suborder Streptomycenae. These include the genus Streptomycetes, which is a common soil bacteria. These produce they can actually produce this substance called jasmine, which gives the moist earth its odor. If you've ever waited until it rained and then went outside and smelled earth, smelled soil, and it has that very distinct smell, that's jasmine. That's due to the bacterium Streptomyces. That gives the earth its earthy smell. And they produce a vast variety of antibiotics. So I hope that gave you a very nice overview of the different microorganisms on our planet. Again, I just touched on a few dozen here, but you should know that there are thousands of microorganisms on this planet of the prokaryotic variety in the domains Archaea and bacteria. These are just a select few I took to show you a good survey of all the myriad of diversity there exists. Anyway, I hope this helped. I hope you understood some of it. I'll make it clear when it comes closer to the exam what to focus on and what not to focus on. I do realize it's a lot of information, so hang with me. Just try to appreciate all the variety and diversity there is in life, and we'll just pick up here with Chapter 12 next time. Thank you, and I'll catch you next time.